Can everybody see the slides? Okay, fantastic. Well, thank you very much, Rochelle, and thank you, Mike, for having me present today. And thank you, everyone, for coming online this morning. And it's an honor and a pleasure. So, let me a little bit about me is I was in Vancouver for 20 years, and I actually miss it. All four of my kids were born in Vancouver, so. And so still have some ties in the Faculty of Pharmaceutical Sciences and ties with particularly with Mike and some of the amazing work that he's doing at the Prostate Centre. And so what I'm going to share with you today is really a 10-year collaboration that Mike and I have had. And in fact, two of the folks that are on this presentation, Dr. Jacob Gordon and Ankar Meeta, are graduate students that we co-supervised. And Mike was kind enough to mentor them through to the finish line when I moved over to Saskatchewan and was dean here for the last 5 years. Finished my role as dean, but I've been able to maintain my research throughout the time, and I'm really excited to be back involved in the work that we're doing around cholesterol and castration-resistant prostate cancer. So the title of my talk today is Scavenger receptor SRB1 as essential component of steroidogenesis and castration-resistant prostate cancer. But what I really want to talk about today is, the relationship between cholesterol and the development of castration-resistant prostate cancer, and how actually we feel that being able to control cholesterol when cholesterol is coming into the cell or into prostate cancer cells, and how it's actually made inside a cancer cell, how if we can block that we think that we might have a nice new novel strategy to go after this castration-resistant prostate cancer. So the next slide, if you see here, I always like to do this, is the take-home messages from today's talk. And many of these things you already know. If you're afflicted with prostate cancer, as I always say, patients sometimes know the best about what they're going through, and so you probably have read up on many of these different things. But some of the take-home messages from today are that prostate cancer feeds off testosterone to grow. We all know that, and depending on the literature, and I'm going to have my little cursor here, so hopefully you can see me follow along. Between 10 to 25% of prostate cancer patients treated with hormone therapy to block, testosterone production, namely ADT, tumors eventually come back despite the therapy. And so the question was, how is it possible for prostate cancer to grow when its testosterone appears to be cut off? And in fact, there was a bit of a misnomer, as many of you know, probably about 12, 15 years ago, that when first castration-resistant prostate cancer was first seen, we didn't think it was, castration-resistant prostate cancer. We thought it was androgen independent. And, the community was actually wrong, because as it turned out, these castration-resistant prostate cancer cells, and thus the name, were actually still making their own androgens and were growing. And what we've seen is that cancer changes cholesterol, which is a building block. And many of you know cholesterol from cardiovascular disease, into testosterone, which feeds its growth. And there has been now a tremendous amount of research done that using cholesterol-lowering agents called statins. And as you well know, statins have been very successful in lowering your bad cholesterol, or LDL cholesterol, to help with cardiovascular disease. There has now been some really interesting research, and we're going to show you some of the work we've done, which has shown that these statins cut off cholesterol's pathways to prostate cancer cells, kind of starving the cells and stop growing. And in fact, what we can do is prevent cholesterol from being made inside the cell so that it can't convert into testosterone, and therefore limit these prostate cancer cells from growing out of control. The other thing is that cholesterol itself is needed for all different aspects of cell growth, not just prostate cancer cell making of androgens. And so it's sort of a double-barreled approach, that if we can shut down cholesterol in prostate cancer cells, that not only that they won't make testosterone, but it will actually cripple its growth. And halfway through the presentation, Mike and I are going to show you what we call a new route to block cholesterol. And we think this combination of preventing these prostate cancer cells from making cholesterol and blocking cholesterol from being recruited outside prostate cancer cells into the cell, this combination we think is going to be very helpful in dealing with castration-resistant prostate cancer. So the next slide is a summary, recent paper that came out. And again, many of you know this, but just to set the scene, prostate cancer continues to be one of the most common non-skin malignancies in men. US numbers are 174,000 new cases estimated per year, and really second only to lung cancer. And in this paper, they talk about that 90% of the prostate cancers are confined to the prostate and adjacent tissues, and that the prognosis is quite good, particularly because we have some great strategies like surgery, radiation, drug therapy, and surveillance. But it's really this last 10%, which is this metastatic disease, this spreading disease. It's incurable, and that causes this castration-resistant prostate cancer. These numbers vary, colleagues, and I would say to you that unfortunately, these numbers are growing. I've read papers now and looked at recent studies that as high as 25 to 30%. And so this has become really the major focus in the last number of years of many research labs and many clinicians on how are we going to treat this castration-resistant prostate cancer, because as our ability to affect this kinds of non-castration-resistant prostate cancer wanes, that the ADT therapy works for a while, but after a while its response wanes. And then these cells become castration-resistant. How do we deal with this? And so this has become a really big story in the last number of years. This slide here is a very nice slide. It may be a little blurry, but it's a nice slide because it shows that, again, the feeding of testosterone, okay, this little guy down here, T, that you get in your things, it comes from the testes and it comes from the adrenals. And cholesterol, this molecule here, is a fundamental building block that ultimately gets transformed to testosterone down here. And we just feel that if we could find a way to block this, prevent it from being converted to testosterone, prevent it from being even recruited into the cell or being utilized, that might be an important strategy to help us deal with castration-resistant prostate cancer. So what is cholesterol? So cholesterol is really multifunctional. I mean, it's involved in bile acid and vitamin production. It's involved in all your cell membranes. And of course, it's transported in the blood circulation by lipoproteins, VLDL, low-density lipoprotein, and HDL. And as you may remember and might know, cholesterol, there's good cholesterol and there's bad cholesterol. And good cholesterol is this HDL, lipoprotein cholesterol, that's high because it can be taken out of the bloodstream very quickly and processed. While LDL cholesterol is the bad cholesterol that stays in your bloodstream and forms atherosclerosis. So cholesterol, as you know, in cardiovascular disease, is an interesting player. But cholesterol is also fundamentally used in so many different things in the cell, and we do need it to live. We just need to regulate it. This shows inside the cell, a little bit complicated, but what I want you to see is the following. You got cholesterol here in the big red. Above it here is an enzyme that when we block it with statins, these cholesterol-lowering agents, we prevent cholesterol from being made. But also, cholesterol is brought into the cell by this LDL cholesterol and HDL cholesterol through an LDL receptor and this thing called scavenger receptor, receptor B1. And this, we'll talk about this in a moment, because what happens in castration-resistant prostate cancer is these cancer cells say, hey man, we'll find any way we can to get cholesterol so that we can convert it into androgens so that we can grow out of control. And so what we try to do is block cholesterol from being made inside the cell and block cholesterol coming into the cell through these receptors. And if we can do both, we might have an interesting way to deal with this prostate cancer. So keep this in mind as we talk about the work that we're discussing today. And this throws you inside the cell a little more in detail. So this is just a generic cell. This first box that I'm showing you here is cholesterol that's actually made inside the cell. So we have the ability to make cholesterol ourselves inside cells. And that's one thing you need to keep in mind. We have these transporters, as I just mentioned in the last couple of slides, that bring cholesterol in the cell from outside that then can be turned into testosterone. And we have actually mechanisms inside the cell that can actually remove cholesterol, these efflux transporters. I won't talk much about this today, but this is another way in which we might be able to get rid of cholesterol so that it can't be converted into testosterone and then utilized in castration-resistant prostate cancer. Now, there is a relationship between cholesterol and high-grade prostate cancer, and there are lots of papers over the last 20 years, colleagues. But here's a paper that I just pulled out from 2018, which the bottom line is that they suggested that there is a relationship between high total serum cholesterol and high HDL were associated with increased risk of high-grade prostate cancer, supporting a cholesterol prostate cancer link. So there's been a lot of anecdotal evidence suggesting this, and that's why Mike and I and many others have said that, wow, investigating what's going on with cholesterol and its link to prostate cancer might be very important to look at in greater detail. Furthermore, there is a lot of relationship if you delve deeper between cholesterol and prostate disease. So what we're showing you here, the bottom line is that there is high levels in the bone harboring prostate cancer metastases. And in these bones, in these prostate cancer bone metastases, there's a high expression of the LDL receptor. So that's where LDL cholesterol is taken up by the cell. This SRB1, this scavenger receptor that we're going to talk about in a bit, that takes HDL cholesterol and brings it into the cell. And this HMG-CoA reductase. This is the enzyme that is used to make cholesterol. And so there's, again, this great relationship between prostate cancer metastases and the increase in the high expression of these different processes here. So again, a relationship between cholesterol and prostate cancer. This is a beautiful slide from 2014, which actually shows visually cholesterol and cholesterol ester and lipid droplet accumulation in aggressive forms of prostate cancer. And so what I show you here is, here's the normal lumen, here's low-grade prostate cancer and a high-grade prostate cancer and a metastases. And you see all these little dots here? And you can see it here when we stain it. And look at it here in fluorescence. Look at that. That's the accumulation of cholesterol and lipid droplets that you find in the high-grade prostate cancer. So again, big relationship between cholesterol and prostate cancer. Now you might ask yourself, well, why is these prostate cancer cells wanting to get cholesterol? Well, not only is it important to use cholesterol as a building block to make testosterone so that these prostate cancer cells can grow out of control, but cell growth itself needs cholesterol for all sorts of different things in growth. It needs it for cell receptors, it needs it for steroids, it needs it for bile acids. Cholesterol is a fundamental building block required in order for a cell to grow and to replicate. And what happens in any cancers, but in particular in advanced prostate cancer cells, they need large amounts of cholesterol to grow out of control. And that's the problem with cancer. Cancer is unregulated control of cell growth. It's not regulated. And so it grabs all this cholesterol in and starts to grow wildly out of control. And so our thinking is, if we can, again, this double-barreled approach, block cholesterol, reduce it, tamper it down, then maybe we can start to control that prostate cancer. So the progression to castration-resistant prostate cancer, this is a nice figure from 2013. Androgen production of adrenal glands play a role in prostate cancer. The overexpression and changes in the androgen receptor play a role. And so we've got to find a way to control this. And get around these tricks that these cancer cells provide. And I show you this cell not to freak you out with heavy biochemistry, but I just want you to understand the complexity of cholesterol. So cholesterol, the molecule up here, gets converted all through all these different pathways and different components, and ultimately makes testosterone here. And so if we could either prevent cholesterol from being here in the first place, or block its pathways so that it can't make testosterone, maybe we have a way in treating castration-resistant prostate cancer. But the reason why I show you this slide is because it's complex, and there's ways in which the cancer cell can trick us and find alternative pathways to make testosterone. And so that's why this is such a difficult disease to treat. And really, we've got to work very hard at it. These are two important papers that came out about 12 years ago, because up to prior to 2008, I would say that the community was fooled. When we started to get resistance to castration, that when castration occurred and the bloody tumor grew back, it was really believed that there was some kind of androgen independence going on, that somehow this had nothing to do with testosterone or androgens being made. But these are two important seminal papers, and the one on the bottom is actually from the Prostate Cancer group in Vancouver. These are two seminal papers that said that no, actually what is happening is these castration-resistant prostate cancer cells are making their own testosterone. They're making their own androgens. And that was a really important discovery, because then it said to us, okay, well, why is that occurring? And what I want to show you today is that building block cholesterol plays an important, very important role in the cell making its own testosterone and its own androgens. So about 10 years ago, a very important drug came out of the market called abiraterone. And this abiraterone inhibits androgen production in a number of different sites. And one of the key places that it affects is cytochrome P17. And where that is, and I'll show you this in the next slide, that's this little guy right here. So remember, I showed this to you before. Cholesterol is being made. It either is being brought into the cell by these transporters, by this LDL receptor, or the scavenger receptor, SRB1, or it's being made by this enzyme, but then it gets converted to androgens. Well, abiraterone blocks this. So therefore, the cholesterol cannot be turned into the androgen and proliferate. And again, here's showing you the bigger picture. We're blocking this enzyme here so that ultimately it cannot make this testosterone, okay? And in fact, the data from this study was actually quite exciting. It showed that there was actually some improvement with abiraterone. It was proved, almost 10 years ago now, in April of 2011, and there was some improvement with using this drug by blocking this particular enzyme. However, it obviously still things were happening. Cholesterol was still being made. Cholesterol was still being brought into the cell. So even though we blocked a pathway of turning cholesterol into testosterone with abiraterone, it wasn't a full recovery at all. And that's why we got thinking, is there a way to just block cholesterol itself? And so this is a slide which shows us a number of different statins, and Simvastatin is the one we're going to talk to you about in a little bit. But these are different statins that are used to treat cardiovascular disease. It blocks this enzyme which makes cholesterol. And there are a whole slew of papers that have come out over the years that show that statin use is associated with reduced incidence of aggressive disease and decreases prostate cancer specific mortality. So if we could block the production of cholesterol inside the cell by using these statins, maybe we can cut down on the aggressiveness of this castration-resistant prostate cancer. Furthermore, there was a recent paper that was published by Jacob Gordon, our former graduate student, and Mike is on this paper, and this is a multidisciplinary retrospective paper that was published in 2018. And what they showed here is that statin use and survival in patients with metastatic castration-resistant prostate cancer, when they were on other drugs after docetaxel failure, was actually, they saw an increase of a 30% PSA decline. So they actually saw that statins may have a potentiation effect. But the problem here was, it's not a problem, this was a retrospective study. And so the comment that was made at the end of the paper, rightly so, was a prospective validation is warranted. What happens if you directly treat with statins and see what happens? So this was embarked in the lab, and Jacob and Ankar, the two graduate students, were the main focus on this. And you can see the whole gang was there, and Mike was the senior author, and I played my role. And what we did here is we inoculated mice with LNCAP cancer cells. Then we castrated the animals after tumor growth. And then we started to treat with simvastatin, one of these statins that blocks the production of cholesterol being made. And then we looked at the development of the castration-resistant tumor just to see what would happen. And the results were really exciting. And what we showed is that the statin reduced the tumor growth and the castration-resistant progression. And here we're showing you the tumor growth as a function of weeks after castration. Here is the control group. And you can see the progression has been reduced. You can see that the tumor volume has been reduced. And the changes in percentage of PSA has been reduced with the greater with the statin versus the non-treated control. And also very important, colleagues, was that the simvastatin reduced testosterone and dihydroxytestosterone production in these cancer tumor xenografts. And that's what we're showing you here. Look at how you see the tumor with the statin is decreasing it. So what we're saying here is the statin is reducing the size of the tumor, and we see the reduction in the amount of testosterone being made by the tumor. And so there's that link that we're cutting off the production, the cell production of cholesterol, and that's leading to the reduction in the tumor, of the tumor size, but the reduction of the cancer tumor making the androgen, making the testosterone. But you know, life is not easy, and life is not fair sometimes, colleagues. So the cell has a way to compensate. So what happens is that the statin might block the production of the cells for making cholesterol, but what it does do is it increases the expression of proteins that can bring cholesterol from the outside of the cell into the cell. So in fact, that's what's happened here. So the cell says, all right, you want to block cholesterol from being made inside the cell? We're gonna get around you by bringing cholesterol from outside the cell. And so this SRB1, this HDL cholesterol protein that brings cholesterol into the cell from outside to inside, is actually induced. It goes up. And that's what we're showing you here with statin therapy. So it compensates. And that's why this is such a tough disease. And even though statins work to a certain extent, it isn't the whole story. So these scavenger receptors, there's a whole slew of them. And I'm not going to go into the details because I could spend a whole talk on them, about them. But there are a whole variety of them that are used for they're surface proteins that can bring lots of different things into the cell. And this is the structure of it for any biochemist that really wants to get a good understanding of how it actually works. But this is the important slide here. And what it is there it is, this protein now, which brings an HDL cholesterol into the cell, then this cholesterol can be turned into testosterone. So even though we block, whoops, let me go back. Even though we block cholesterol from being made by the cell itself with a statin, the cell says, hey, I'm going to bring HDL cholesterol into the cell, and I'm going to make testosterone that way, all right? And so this HD interacts with lipoproteins, HDL cholesterol, and that's what happens here. What's very interesting is a postdoc in our lab back in 2010, Carlos Leon, showed that this SRB1, this protein, so if I go back, this protein here that brings an HDL cholesterol on its own is overexpressed or has an increase of expression in these tumors already. So it is a pathway for castration-resistant prostate cancer tumors to get cholesterol. So cholesterol is being brought in, not in from the outside, as well as being made inside the cell itself. And so we're saying we've got to find a way to block all these pathways. And here's again showing you in castrate-resistant prostate cancer cells that there's an increase in the expression of the protein as well. What we find is that if we can silence or reduce this SRB1, this HDL protein, we can actually decrease the PSA levels and the viability of these prostate cancer cells, these C4-2 cells. And that's what we're showing here. Here is the decrease in the PSA concentration, and in day 3 and beyond, you can see the decrease in the cell viability. So what we found is that blocking this SRB1 from bringing HDL cholesterol into the cell also reduced cell viability and PSA levels. So this really brought us to our hypothesis, which is if we can inhibit SRB1 by attenuating the androgen receptor signaling and decrease cholesterol availability by de novo synthesis, that maybe we have a way of dealing with CRPC, castration-resistant prostate cancer. And you'll see near the end, combining it with statins, drugs that can block the cholesterol from being formed itself inside the cell, this combination might be a really new novel way in how to attack castration-resistant prostate cancer. And in fact, last year we published a very important seminal paper in cancer research. This was the formation of Jacob's thesis, and again, the whole gang is on there from the Prostate Centre and colleagues around different labs. And it was a very important paper because it was one of the first papers to show that there was a relationship between this scavenger receptor required for steroidogenesis and even non-steroidogenic cholesterol metabolism in prostate cancer cells. And so what we're showing you here now is that we can actually affect this expression. We have tools to be able to affect this expression prostate cancer cells. So siRNA is small interfering RNA, and what that is a genetic way of blocking so that the SRB1, the protein, cannot be made. Its expression cannot be made. And if it cannot be made, then it cannot bring HDL cholesterol into the cell. And so what we're showing you here, and this is the important slide right down here, is that when we use this technique, we can reduce the expression of this SRB1 in these castration-resistant prostate cancer cells, these C4-2 cells. And what that does is by silencing or reducing the presence of this SRB1 protein, this HDL cholesterol protein, we can alter the cholesterol metabolism. The cholesterol uptake from HDL is decreased, as we see here. The cell viability itself is decreased. We can actually then reduce the cellular androgens inside the cell. So you can see the testosterone is decreased. The dihydroxy testosterone is also decreased. So we're not only affecting the amount of cholesterol coming into the cell, we're affecting the cell viability, its ability to grow and survive, but the androgens that it needs are also been decreased. And we're able to affect its androgen activity as well. Here, the PSA goes down, and here you see the PSA concentration, its expression goes down, and a couple of other important factors as well. What's also really important, remember I told you that cholesterol is really important just for the life of the cell itself. Yes, it's extremely important as a building block to make testosterone, which is required for castration-resistant prostate cancer, but blocking cholesterol itself can affect the cell from actually turning over, from replicating, from going through its cell cycle. So SRB1 silences the cells, that induces cell cycle arrest. So like a cardiac arrest, it actually blocks the ability of the cell to turn over, to cycle. So we're not only affecting the testosterone being produced inside the cell, we're affecting the cell itself. And so that double-barreled approach that I talked about at the beginning, the take-home message of this work. What it does not do is there's a thing called programmed cell death called apoptosis. And we find that we're not programming its cell death. We're doing something very specific here. And that's very important to recognize. And it induces growth arrest. So it's almost like a cardiac arrest that we're giving, like you think about with heart disease, we're doing to the cell. We're arresting it. We're not letting it propagate and grow this cell because we're shutting down the ability of the cell to get this HDL cholesterol, this cholesterol from outside the cell to come in. So what are the take-home messages from this? You know, the reduction of testosterone and DHT accumulation, as well as corresponding decrease in the androgen receptor pathway, and we can arrest the growth of the cell by blocking and silencing this SRB1. So the next step is coming up with a therapy now to do this. It's all nice and great to have this kind of cool data inside the cell and even in that, but can you actually have a therapy or a combination therapy to deal with it? So there's a molecule out there called, a small molecule called SRB1, which is called BLT-1. This was actually a product that was going to be used in cardiovascular disease, but actually didn't work very well. And so what this does is that it actually prevents cholesterol from HDL cholesterol to be taken up into the cell. So what we did is we said, okay, if we use this molecule, this inhibitor of this protein, of this SRB1, what does it do to cholesterol uptake in PSA? And you can see it actually reduces cholesterol uptake and it decreases PSA concentration. And this is where it becomes really exciting and interesting and will be hopefully some of the future work that Mike and I are going to embark on. We've got one grant from Prostate Cancer Canada to start looking at some of this, but Mike and I have also submitted some other grants that we're waiting to hear about. There's been a delay, unfortunately, because of COVID-19 that we hope to get money for. But what we're showing you here is we've taken the BLT-1 molecule, this molecule that blocks HDL cholesterol from going up in the cell, and statins, which are, again, if you remember, the molecule that blocks the production of cholesterol that's made inside the cell. And when we combine them together, so we're blocking cholesterol coming into the cell and cholesterol that's being made by the cell, what happens? And what we show is a combination synergistic decrease in PSA secretion. So here is the BLT alone, decreasing PSA. And look what happens when we use them both together in different combinations. Look what happens to the PSA concentration. It goes down significantly. So this is very exciting because now what we could do is combine these two molecules, these two different pathways to try to shut down, all right, cholesterol coming into the cell and ultimately being transformed into testosterone. And we can also block the cell growing out of control because it needs cholesterol in the first place. This slide now shows you some of the pharmacokinetics and some of the work we've done with animals with BLT very early on. This just shows the BLT concentration as a function of time when we give a PO dose to an animal, and you can just see these are the kinetics. But more importantly, and important to know, is the tumor volume changes of the BLT alone. Look at this red, this blue line says this is the volume does decrease compared to the control. The body weight, that it's not really significantly different, a little bit lower, but not too much toxicity. And what happens to the percentage below the vehicle. So what is our conclusion, colleagues? Is that we believe cholesterol is essential for several cellular processes in steroidogenesis. And that if we can inhibit cholesterol availability, it may be a viable therapeutic strategy alone or in combination for the treatment of castration-resistant prostate cancer. And I go back now to the slides that I gave, the slide I gave you right at the beginning, the take-home message. Again, we want to cut off cholesterol from being involved so it can feed the growth of prostate cancer. And we're doing that by blocking cholesterol, all right? But we're blocking it in a number of different ways. Not only the production that it has itself, but also by the way that it comes in through this HDL cholesterol mechanism. And so we think of using this, what we call this double-barreled approach, that we might be able to deal with castration-resistant prostate cancer. Now, let me tell you the other exciting thing about this, is that by having this extra tool in the toolbox, we might be able to go back and use it with other therapies that are already on the market, like abiraterone and others. And maybe using an SRB1 inhibitor with abiraterone, or using statins and SRB1 and an ADT therapy together might work. One of the biggest problems with those kinds of complicated therapies has to do with the timing of what to give to patients. And of course, they're always worried about the toxicity that each different drug brings. But again, it provides us with new tools and new therapies to try to deal with castration-resistant prostate cancer. This is a summary from really kind of independently of what I've talked about today. But the reason why I put this in as other key factors is that this is a very complex and moving field. The transition to castration-resistant prostate cancer really affects androgen-responsive genes are expressed despite androgen deprivation therapy. And that we then learned that the adrenal is really a major source of androgens and androgen precursors for men with CRPC, and that there are major androgen precursors, okay, all these other components that are involved in androgen synthesis and enzymes that play that role. And we even have found now that there's a shift from androgen receptor to the glucocorticoid receptor in some of these CRPC tumors. And so when I show you this slide again at the end, what I'm saying to you is that, yeah, we cut off that, we deal with the testes, we do surgery, we do radiation, but the adrenals start to kick in. And even if we start to cut off and block different things and different pathways, other pathways start to creep up, other enzymes seem to creep up, ultimately making testosterone and dihydroxytestosterone. And so we still have a long way to go, but our feelings are that if we can start to attack things right here at the cholesterol molecule, kind of the fundamental building block that ultimately leads to testosterone, that we can ultimately have something that is special in trying to treat castration-resistant prostate cancer. You can't do this work without a village of people and grant funding. These were the number of people that were on Jacob's PhD committee and that we've worked with over the years that did a lot of the analytical work, that did so on. Mike is a humble man, but I'm going to give a major shout out to Mike, because Mike's lab, once I moved to Saskatchewan and decided to do some stupid administrative stuff, and I'm happy I'm done with that now, Mike really carried the ball as we finished some of the work, because really the early work with SRB1 was done in my lab at UBC back in 2010 and 2011. But to take it to the next level, I really needed to get someone like Mike and the Prostate Center to help us out. And Martin Glebe and Mike Cox and those guys have been fantastic in helping us, carrying us to this point. And we hope now to continue the work moving forward. So I thank you very much for your time, and certainly open for questions or comments. I don't know if there's been chat. I've been trying to not pay attention, but I think we have plenty of time for questions or comments as well. So thank you very much.